All right, so it is 2 o'clock, so we will get started. And what we're going to go over today will be the last material that will be covered on test 1, that is this Friday. I'm still setting up test 1 to run through our proctoring service. Just have to wait for like validation. So I can't really tell you the specifics of how everything's going to work yet. I'm going to play around that hopefully by Tuesday. Hopefully they'll get me all validated and all that. And I'll tell you exactly, how to log into the test, if there's anything special you need to do. But like I said, I will give you more information for that on Tuesday. But let me launch our PowerPoint for today. Today is not Friday, it is Monday, the 14th. Okay. And change this. All right, now we are good to go. So what we're gonna talk first is about modified amino acids. So we talked about 20 amino acids are encoded by DNA, and that's true. And those are the amino acids we need to know the names of, need to know the structures of, and we need to know the pKa's of them. But in your body, there are hundreds and hundreds of more modified amino acids. These I won't ask you to memorize because there's way too many. But what you should see is when you look at these modified amino acids, it'll tell you what the modification is right in the name. O-phosphoserine means we have a phosphate group on our serine. Gamma carboxyglutamate means we have another carboxyglut on the gamma carbon of glutamate, so on and so forth. So my first question to everybody here is, again, a little thought experiment. Big picture, big brain thinking. We do not encode for modified amino acids in DNA. We can't. Well, I guess we could, but we don't. Let me say that. So how do you think inside the cell amino acids are recognized to be modified? And if you have an idea but don't want to like type it out and you want to talk, feel free to raise your hand as well. But I'm just seeing what people think. And one sec, I'm gonna turn on my fan because it's getting hot in here already. Their shape. Shape of what? Be a little more specific when you say their shape. How do we know to modify a specific amino acid? And I got a private message from a student that says their shape, which is along the right lines. Just want a little more explanation. They don't match any of the 20 regular amino acids. What do you mean by that? Because the way we modify these amino acids is that we modify one of the 20. Like if we look at our 5 examples here, they're 5 amino acids. They have just been modified. So we need something to build from, and we always build from one of our amino acids. But how does the cell know to modify one amino acid and not another? How do I know I want a specific lysine to be gamma? Some sort of tag. Maybe explain how a tag could be used. Doesn't RNA modify some sequences to swap base pair additions? mRNA can be modified, yes, after it's been copied from DNA, and it does go through a lot of modifications, but I don't think the mRNA itself can really dictate in the code itself that this amino acid needs to be modified because it's under the same constraint of DNA having to be read by a codon table. Name me the part of the body it's in. So let me modify that and say it depends on what cell type it's in, whether something will be modified. But how do we know? Not every single lysine in a specific cell will be an acetylysine. Not every histidine will be methylhistidine. So I'll give maybe a minute more on this. All good thoughts. Again, never be afraid to throw out an idea, because this is how you learn, right? By stretching your mind. Just like lifting weights hurts your arms and your legs, thinking will hurt your brain to only make it stronger. Is it because the receptors change, like the shapes of them? So again, we have another person with shape. So let me just mold that answer, because it does have to do with shape, at least most of the time, right? So the way these amino acids become modified is that some enzyme has to modify them, right? An enzyme, even though we haven't talked about it yet, an enzyme is just a protein that does specific tasks. So I guess, I don't know, there's my enzyme, right? And an enzyme works on a specific portion of something else. It could be DNA, could be a lipid, could be protein. Here, we're working on other proteins. And enzymes, proteins, all of that, your cell, it's not smart in that it doesn't have conscious thought. The way anything reacts in biochemistry is all through geometry and electrostatic interactions. That is geometry and charges, right? So a protein that adds a methyl group, you will need an enzyme that specifically fits whatever needs to specifically fit not only this histidine, but what is around it. So somebody said like a tag. That's kinda correct. I want a little more explanation on that. It's not quite lock and key. We'll learn a little bit later. It's more induce fit. Lock and key is probably not 100% correct. For those of you not quite sure what lock and key means, lock and key is that an enzyme binds specifically to its target, like a key fits into a lock. We'll learn that that's not quite how an enzyme works. It works more through an induced fit, but it's the same idea where an enzyme recognizes something based on its geometry and its charge and binds to that. So yeah, there's like some like tag or some geometry that will recognize that specific amino acid and add the functional group to it. Some proteins can add like a phosphate to a lot of different proteins. So like there are just there are kinases that can phosphorylate multiple things. There's also like for example methylases. There are some enzymes that only work on one specific protein. So, but that's kind of getting into the weeds there. But basically, you know, when we're thinking about interactions in any chemistry for the most part, even though I like to talk about that things know what they're doing, things have no idea what they're doing, and it's all physics. This is 100% physics based on geometry, charge, polar, nonpolar. So yeah, any questions about that idea? I myself love biophysics. Not everybody does, so I'm not going to get too in the weeds of that. What exactly was the answer? Shape and charge. They recognize the geometry and the charge groups around the amino acid, right? So if you want to think of it like lock and key, that's fine for now, where a specific protein's like a key that will fit into an enzyme lock, and then your chemistry will happen. That's an easy way to explain it. Not 100% like that, but it's a fine, fine analogy. Anything else? And I wasn't laughing at that question. I was laughing at myself for having this 7-minute discussion on geometry and then like, what did you say? Yeah, I can be a little more verbose than I need to be. All right. Oh, okay. So one type of modified amino acid, and I'm sorry it's cut off a little bit on the bottom there, is our friend GFP. GFP, green fluorescent protein, was, I believe, originally found in jellyfish. And what happens to GFP is that you take 3 amino acids, serine, tyrosine, and glycine. And then you do a chemical reaction on these using an enzyme, and you make what's called a fluorophore. Again, I know this is cut off. It's just showing serine, tyrosine, and glycine combined. I'm not going to ask you to draw this fluorophore. Just get the general idea of what I'm talking about here. What a fluorophore is that if you hit it with a specific energy of light, it will shine light back at you. GFP, as by its name, shines green light. That was really the first one discovered. You can do the whole rainbow now, as shown here. You can basically get any color you want from a protein. You can do these weird experiments where like you put GFP in every cell. So like in a rabbit, you put it in every cell of a rabbit for some reason. Now you have a green rabbit when you hit it with light. Fruit flies, worms, mice, basically examples of everything. Here's a cell. That's mainly what it's used for. But so 2 questions again, just get us thinking. Why would you ever want to use GFP in an experiment? And the answer is not just to make a green rabbit because you can. And how would you actually attach GFP to a protein inside of the cell? How would we go about that? To tag, mark something to track a certain protein. Yeah, so the 2 big reasons are, they're not only, there are more reasons. To like tag a protein. Basically, one of the ways you can do it is you want to see where proteins are located. So as shown in this picture above, we have 3 different proteins that are labeled: a green protein, a red protein, and a blue protein. And just by tagging them, adding these markers, we can see where in the cell they're located, and if we have what's called co-localization. Co-localization just means 2 proteins are like near each other. So yeah, you can see if a protein's actually in a cell. You can see where it is in the cell. You can do some things like track protein speed to see how fast a protein moves using this. But yeah, mainly it's just tagging purposes. So let's say I discovered a new protein. Let's call it the Tamusa protein. I discovered this new protein. I want to know inside the cell where it's located. So I'm going to put a GFP tag on it. And that's how you would actually call it. You call it a tag. How would I attach GFP to my protein of interest? We kind of have already talked about this before. Hint, hint, wink, wink, nudge, nudge. Yeah, use a vector to put the DNA that makes GFP into the cell. So yeah, let me just expand on that, right? So you have your protein, the Tamusa protein, and you get the Tamusa DNA. So you get this DNA sequence. And generally what you do is you add GFP somewhere, right? You can add it at the end of your sequence. You can add it to the beginning of your sequence. If you're really brave, you can add it to the middle. There's pros and cons to doing, well, there can be cons, I'll say that. Depending on where you add it, it can change how the protein functions, right? Because this molecule is a big old protein, right? So if Tamusa was this size and you want to study it with GFP, you're basically saying, okay, that's GFP right there. That's Tamusa. I can't spell. Yeah, Tamusa. You're basically saying, oh, these are the same things, which might not always be the case. So you usually want to do some experiments where you put GFP in your DNA sequence. Just make sure that your protein is still doing what you think it should do. Is it still doing the correct chemistry? Is my cell still alive with this GFP version in it? So does that make sense about GFP, or any questions about tagging a protein or GFP specifically? Let me get rid of all my scribbles. So it's not quite a question. If GFP does somehow change the protein, what are the alternatives? There are other fluorescence tags out there. So you can see if they'll work. They're different sizes. So that's one thing you can do. You can also, I don't even know if I want to say this because I don't know if it's right. What are the alternatives? I don't know off the top of my head what they're called, but there are a lot of different fluorescent proteins. There's like a whole study of research of just creating new fluorescent proteins. GFP is just like one of the first big ones. But since then, people have just found, they go through nature and just see what are fluorescent compounds that we can use for this. Yeah, I'm trying to rack my brain if your fluorescent tag wouldn't work, where you could find it. And inside of a cell, doing this experiment in vivo, don't know of a great alternative other than just trying different tags. Usually, it will be probably inert, because if I draw my DNA, right? Let's say it was your job as a student to put GFP on a protein and make sure it worked. The first place you would put it on the N term or the C term. And if that doesn't work, I would just start putting it in random places and seeing if it would work. If that didn't work, of course, I would move to a different fluorescent protein with different size. Good question, though. Any other questions? Sorry, I couldn't give you the best answer on that. I don't have those fluorescent names off the top of my head. Yeah, it's interesting because when I went through grad school, we were a structure lab, so we cared a lot by the structure. And most people who do that type of work you're talking about, they don't always think about what a giant protein tag would do to the structure. So just have to be aware of it. That's it. Yeah. So in our body, right, we have amino acids that are also biologically active. And you've probably heard of these, like dopamine, histamine, maybe GABA. So we have our neurotransmitters, and look what they're made out of. They're made out of amino acids. Again, I'm not going to ask you to recognize, I'm not going to ask you to draw histamine or dopamine or recognize those for me. But something I might ask you is I might say, here's a picture of serotonin. What amino acid do you think serotonin comes from? And those of you who have thought about neurobiochemistry, what does serotonin do, or what does a lack of serotonin cause? Anyone knows that? Yeah, if you don't have enough serotonin, you get depression. That's one of the things. I'm sure there's a lot more. I am not a neuroscientist by any means. But let's see how well we've been studying our amino acids. What amino acid does that look like? What amino acid has a double ring structure? That's our friend tryptophan. Yes, tryptophan is the only amino acid that has fused rings like this. And how can we remember that? Well, the one-letter code for tryptophan is W because it has a double ring. That's actually why it was called W by the person who created the one-letter code. And just a random thought because I'm thinking about it. If you'd like to look it up, Margaret Dayhoff. Person who created the one-letter code is Margaret Dayhoff. She was like I think she was a dual PhD maybe, computer scientist / biochemist in the 50s. But yeah, you can read all about why she I might have talked about it in my videos, why she picks the one-letter code. But yeah, that's tryptophan. And again, this slide is amino acids just aren't for proteins, but we can modify them. We can use them for different purposes inside the body, for signaling purposes, ligands, all that good stuff. So let's briefly talk about primary structure now. So we're slowly getting into proteins proper. And proteins have 4 different levels of structure. We can talk about them. They're called primary, secondary, tertiary, and quaternary. Primary, secondary, tertiary, quaternary. Primary is simply the amino acid sequence. From N-terminus to C-terminus, what is the amino acid sequence? And when we talk about proteins, it's amazing the number of proteins that you can actually make with just 20 amino acids, right? So if you have a protein that's just 100 residues long, using this calculation, 20 to the power of 100, you can make 1.27 times 10 to the 130 different combinations of amino acids. That is probably more combinations than there are atoms in the universe. So 20 amino acids seems to be like a great number to do like whatever we want. However, a tiny fraction of these proteins are seen in nature. Like in E. coli, I want to say there are 3,000 unique proteins roughly. These peptides are between what size? A, B, C, D, or E? Do I have a poll for this? I should. I don't. I forgot to make a poll. So what is it? How big do we think these proteins are? I might have mentioned this in the video. Why are they this size? Yeah, they are roughly between 100 and 1,000 amino acids long. Can anyone think of a reason why they might be that long? Because you need more than 40 to fold. Roughly, yeah, 40 is probably the smallest. Yes. So you need a minimum number to be able to fold to a complete 3D structure, and roughly 40. There's one protein called tryptase that's around 40 amino acids. That's going to be like the smallest amino acid. 1,000, yeah, it's not going to be able to like fold correctly probably that well. And the function, it's just kind of like overkill more or less. Although I say that, the biggest protein that's ever been discovered is in humans, and it's called titin. And I want to say off the top of my head, titin is roughly 24,000 amino acids. So there are exceptions, titin being the biggest. But generally, most of your proteins are going to fall 100 to 1,000 amino acids. So I'm not sure why I'm showing you A chain and B chain here. I forgot what my reason is. Other just to say that, remember, the only amino acid that can form disulfide bonds are cysteine. I know why I put this chart here. Let me move this image up. One sec. Let me get out of writing mode. Okay, move this image up. Delete that question. Don't need it. Okay, and let me get rid of my drawings. The point I wanted to make here is that just because you know the primary structure, proteins are three-dimensional. So something that might be far away in primary structure might still actually interact right next to each other. That's because, primary structure, I think of it as like a long line or a long spaghetti, and this spaghetti folds up into a 3D image. So when it folds, certain parts of the proteins that are far away in structure or in sequence can now come together. And one way that you can help have things come together are through these disulfide bonds. Sulfur will or cysteine will bond with itself via the side chain. So two cysteines will form what's called this disulfide bond, and you can bring different parts of a peptide together, or you can actually combine different peptide chains together through a cysteine bond, or disulfide bond rather. So just knowing the primary structure won't tell you what amino acids are actually interacting inside your protein or are near each other. That's why we need to get the secondary and tertiary structure, but it will tell you what amino acids are in your structure. So that's mainly what that image is for. All right, any questions about anything so far? Now I'll move on. So we do have a question. So disulfide bonds make the structure. They help. They're one of many things, but yeah, they help. They're not the only thing. And we'll talk when we get to secondary and tertiary and quaternary. We'll look at that a little bit more, but they can help make the structure. Right. So studying proteins. If we want to study a protein, usually we want to purify the protein from the cell. What are some pros and cons of studying a protein outside of the cell? And this is an open question. I'll make a pro and con list. Pro, con. So what are some ideas? What would be an advantage to studying a protein? That is a con. Isolated. Yeah, isolated or interactions. So like if you're trying to study like a car, if you saw a car for the first time and you wanted to know what the seatbelt does and you just took the seatbelt out of the car, you wouldn't probably know what a seatbelt does. So you're kind of missing its interaction without like the human being there saying, oh, it keeps the human inside the car. So that is a con. You can't see how it works in the bigger picture all the time. What are any other pros and cons? Pro, you can make a bunch of it. Yeah, you can get a ton. Get A lot of sample. Inside the cell, especially when it comes to like human proteins or any other real protein, if you want to study it inside of the cell, you are usually looking at few copies, depending on what protein, of course. But like inside the cell, we just don't have a ton of copies of everything. While if you isolate it, you can amplify it by using techniques we talked about and get a ton of it. No noise, yeah. Results are from it alone. So if you isolate a protein, you know whatever result you see from that experiment is from that protein because there's nothing else in there. And I'll go with just a con. You already said it, isolated. But one thing experimentalists don't think about is that even if the protein you're thinking about studying really doesn't interact with other things all that much, inside of the protein is crowded. There's about 300 mg per mil of just dissolved stuff inside of a cell. In a test tube, the average concentration of a test tube is about 10 mg per mil of dissolved stuff. So a protein, when you purify it, you are taking it from you're putting it in an area that is 30 times less concentrated. And studies have shown that this, even though all of the things that are concentrated might be inert, this level of concentration differences can affect how a protein behaves. So that is something else to be aware of, is that the results you see might actually not be real as to how a protein behaves inside the cell. Yeah, right. Results might not show how the protein would behave naturally. It can easily fall apart outside the cell. Yeah, denature. Pro, way easier. It is so much easier to study a protein outside of the cell than it is in. You can do a lot more experiments with it too than inside the cell. People are getting better at doing in vivo experiments all the time because you really do want to do in vivo experiments if you can control everything. Hard part is controlling everything. So that's outside the cell. So let's do E. coli now, and I'll just kind of rattle these off. So E. coli, pros and cons. So E. coli, cheap, well studied, relatively fast. To get like E. coli samples, it's less than like $100 to get a bunch of them. And then once if you get one sample of E. coli, you can just make them infinitely, right? So it's like a one-time cheap investment. Now you have E. coli forever. E. coli is a model organism. We have studied E. coli a lot. We know how E. coli works pretty well. E. coli also replicates very fast and grows proteins very fast. So you can get your purified protein. You can go from creating a vector, looking at the vector, getting the vector inside the E. coli, growing that E. coli, purifying a protein in a week. You can go from scratch to done in a week if everything goes well. Con, no post-translational modifications. We talked about this already, the cons of this, post-translational modifications. Maybe not fold correctly either. Also inclusion bodies. It could just put all your protein into like one vesicle and inject it out of itself. You can definitely kill E. coli with having too much foreign protein. And it is possible that E. coli will recognize a foreign protein. and chop it up for parts. Usually from what I've heard though, it'll just put them in these inclusion bodies and they'll get all misfolded and just, it's more like the E. coli knows this is all junk, put all the junk together and get the junk out of me. But yeah, there's no guarantee that it would work. Usually though, what you do if you've never done this experiment before is that you shake the E. coli, right, till they get to a certain density, and then you induce, and then you all time it out. So you kind of like, based on other people's experiment, because E. coli, like I said, is so well studied, that you have a generally good time frame to know when things are optimal. Again, it's a little bit trial and error, but usually you can look up protocols for that, no problem. So my last question here. Lysine. Lysine means to basically break in general. Here I'm lysing a cell. So I'm taking a cell who's happy, adding something, killing it, and all the gooey contents come out. What are some challenges when I burst open a cell or lyse it and have all the goodies come out? What are these challenges I have to overcome? What are the challenges and how do I overcome them? What ideas do we have for that? The goodies touch the outside. Yes. Why is that a problem? Whatever chemical lysis that could denature the protein. That is true. So usually when you, so could denature. So that could be from a number of things. Could be concentrations of salts, right? Could be how you actually lyse the protein. What chemical you could do that. There are also, it could also be, what else could denature your protein, right? So we have like concentrations of stuff like heat, yeah, proteases, pH, yes. So pH, that's easy to deal with, buffer. So when you do this, you make sure your solution's buffered to keep basically the same pH that your protein would be happy in. Air is also one, yes. Air. So to have this, when you are like doing this process, you want to be gentle and not make like bubbles, because bubbles will basically denature your protein, having that like little air pocket there. So you kind of try to cut this open as gently as possible. Heat, keep that on ice. Anything over 4 degrees and your protein will probably denature, except like super hardy proteins. And so generally you want to try to be as careful as possible, but it also depends on your protein, how much of a loss you're willing to take. Because when you're purifying proteins, you're gonna lose proteins, and that's just something you accept. So here on this image, I have different ways to purify protein. Blender. So I used to teach a lab like 5 years ago now, where we would get a protein out of a chicken breast. And our first step was blend the chicken breast. That's a good way to cut open cells. Good way to get it all ready to go. Will a blender make bubbles? Oh yeah, a blender is the harshest way you can do it, but our protein from the chicken breast was in such abundance that it was the cheapest and fastest way to do it. And we still got plenty of protein left over. So it was something that we were willing to do. Here, called sonication. Sonication uses sound to burst open cells. Tiny pestle. Yeah. we just bought chicken breasts from, well, it was in Iowa, so it was from Hy-Vee, and threw that in there with some ice with some other chemicals and just let it rip. Anyways, sonication, that here, sonication is the most annoying thing in the world because you use very high frequency sound to literally bust open your cell wall. So when you do this, you go in a room, like it's supposed to be soundproof, but you can still hear the sonicator work outside the room. And you have on these big, huge earmuffs to try and block the sound. That still doesn't really help. It is super high pitched and it sucks, but it works quite well to just burst open your sound. You can also do like pressure, where you just like burst the cells open with pressure. You can also use chemicals to do that as well, like detergents. But again, that might, depending on what protein, that might ruin your protein. Some proteins, you only have to do it with detergents. Like if your protein's found in the cell wall, the cell membrane rather, you have to use a detergent. If you don't, your protein will denature when you bust open the cell membrane. So any questions about like any of these like pros and cons? Let me clear this drawing. Okay. Now, when you're purifying a protein, we need to be able to know how much of it there is and if we have it at all. One of the easiest way to do it is by aromatic amino acids. And for some reason, that text is cut off. So aromatics, amino acids, W, F, and Y, will absorb UV light at a wavelength of 280. Really, it's only tryptophan that does it. Tyrosine does it a little bit at 280, some at 260. F is like very tiny. It still does it technically, but what you're really looking for is when you burst open the cell and you're going through these purification steps, one easy way to see how much protein you have is just shine a 280 nanometer laser at your sample and see how much absorbs, and then you can do a calculation to determine how much protein that is. Since we have a test on Friday where I'm asking you to know your amino acids, I give you 2 peptides. Which one would absorb greater at 280 based on the 3-letter codes? Would it be peptide 1 or peptide 2? How well do you know your 3-letter codes? Gotta vote for 2, gotta vote for 2. Who knows their 3-letter codes? What is tryptophan? And actually, I probably said the 3-letter codes when I was talking about amino acids because I rarely use the full name. I usually use the 3-letter codes. Yeah, seems we're pretty unanimous. Peptide 2. Trip is W. Let me get my pen. Trip is W. Phe is phenylalanine. F. Tyrosine is Y. Tryptophan. So that peptide 2 is going to absorb way better than peptide 1, mainly because of the tryptophans. That being said, if your protein has no tryptophans, you generally cannot monitor it at 280 nanometers. Also, since Y absorbs at 260, DNA also absorbs at 260. So when you are seeing how much DNA you have, if you're purifying DNA, you have to do a certain calculation that compares the wavelength at 260 versus wavelength at 280 to know if any signal from 280, or sorry, from 260 is actually protein. So that's just a fun fact. I don't expect you to memorize that or anything, but that's just a fun DNA assay fact. So that's relatively simple, but any questions about that idea? Again, light goes in, 280. Absorbance equals protein. Shine light, 280 absorbance equals protein concentration. Yeah, 2 because 1 doesn't have any tryptophans, and the only aromatic is tyrosine, even though it's kind of cut off. So that's tyrosine right there. But yep, you're looking for the tryptophans. Okay, the last thing I believe that we're going to talk about is ELISA, the enzyme-linked immunosorbent assay. ELISA is used along with Western blots, but we're not really going to talk about Western blots. We're going to focus on ELISA. But it is used to determine if your sample has your protein of interest. So I'm going to walk through how ELISA works here. So you have some kind of support, solid bed support right here. And on that bed support, you usually have an enzyme, or sorry, not an enzyme, an antibody. This is called your first antibody because it's the first antibody. And then what you do is you take your sample, right? So let's say you're trying to purify a protein and you want to know if that protein is there. You take your sample that you think has your protein and you put it across this solid support. And antibodies are very specific in that they only bind one type of antigen. What an antigen is simply something that an antibody binds. That's what an antigen is. And so generally your first antibody is specific for your protein and your protein only. So if your protein is present, your antibody will bind. If it's not, nothing will bind. So you incubate your sample with your solid media, and then generally you wash it. So anything that's not bound or not bound strongly will be washed away. Then, after that, you add a secondary antibody. This secondary antibody usually has some kind of enzyme on it. So this antibody, and by the way, antibodies are proteins themselves. This antibody will be added in, and it will recognize either your protein, or what will happen sometimes is that this second antibody will only bind to antibodies that have something like bound to it. And it really depends. But in this image, what we have is we have this secondary antibody binding a different part of your protein. So it's binding a different antigen in your protein. So you add the secondary antibody, you let things bind, and then you wash it away again because you don't want any secondary antibody around to give you a false positive. Okay, once we're at that step, we add some kind of substrate. This substrate will interact with the enzyme that's on your secondary antibody. Then you will create some kind of product. This product is either like radioactive, or it's colored, or it's fluorescent. Some way we can determine that your protein is there, right? That's ELISA in a nutshell. Now, if the enzyme the protein you care about is an enzyme already, what you can do is that you don't always need a secondary antibody. You just add your reagent and just see if it's there. That's rare, but it is possible. It is also possible, but rare again, that your first antibody or primary antibody could have a signal by itself. Again, I've heard about it. I've not really seen it in practice. Mainly the way it's on this slide is, the general way to do it. So is there any questions about the process of ELISA itself? Like why you would use it or what the steps are? Both things you should know by Friday. Hint, hint, wink, wink. not, I have one last question. Just see if you understood everything I said. They could use that for viruses because viruses are made out of proteins. If you're looking for a specific viral protein and that protein doesn't change too much, you could do that. No problem. Yeah, you could use it for COVID as long as you know as long as you have an antibody for that protein and that protein inside of COVID doesn't really mutate. And so when you do that, there are specific proteins in viruses that are more, and blanking on her name now. The head of biology would know that much better than I would. Wise would be able to probably talk circles around me when it comes to this stuff. But basically, in the virus, certain proteins will mutate faster than others. And so if you know of a protein that doesn't mutate all that fast, one, that's a great drug target. Two, you could use it for something like this, where you create an antibody against it. Then you can do ELISA to see if it's there, right? And I thought they did use ELISAs on COVID off the top of my head. I might be wrong about that, but I thought I remember reading that. That's one way, that's like a great detection method. It's not the fastest. It takes roughly a day, but it is a way to do it. Could be faster than a day. I might just be talking nonsense. But last question. When you do an ELISA, when you do ELISA, and my sample does not contain any antigen, or my antigen that I'm looking for rather, what would be the result of your ELISA at the very last step? And why? Yeah, you would see nothing. So if your ELISA, right? If your ELISA is on that surface and you don't have an antigen for your antibody, nothing will bind in the first step. Because yes, antibodies are so specific. They bind to one thing and one thing only. For the most part, it is possible that they'll bind to other things, but we're not going to get into that all too much right now. But they are specific. So if your antigen's not there, then the second antibody will not bind. The second antibody has a signal. So if that won't bind, then you're not going to get a signal. So it will be blank. That's a negative. It's not there. So any questions about that or ELISA or anything? Test on Friday. I'll put a homework up today. This is the last PowerPoint that will be on the test. Wednesday PowerPoint won't be on the test. How long is the test? You have an hour. How many questions? Not sure. Those are usually the general. I have a question about the test. Shoot. Watching during the test. A computer will watch you, and I think I can watch you whenever I want without you knowing. That's a way to make sure nobody's cheating. That's how I was told it works. Is it through Blackboard? It should be through Blackboard, yes. Do you have to download anything? I don't think so. That is one thing I'm gonna be like test driving and just confirming on Tuesday once I get my account all ready to go. But yeah, it should be a link on Blackboard under course content. And the only thing that's like really different is, yeah, I'll double check on that just so I don't say any false information right here. Will I provide you with pKas for side chains? I will not. You will need to know the side chain pKas. Do we have to learn any specific equations? You should know delta G equals delta H minus T delta S. You should know the buffer equations. pH equals pKa plus log base divided by acid. I think those are the two equations we really used in class to solve problems, unless I'm forgetting one. Which I don't think I am, but I'm gonna go through all the stuff we did and make the test soon. In isoelectric point. What I really care about is that if I give you a peptide, You tell me the charge at 1714. I probably will not give you, like say, this is arginine. What's the isoelectric point of arginine? Not too concerned about that, so probably not. You have to know what PI is. You have to know what an isoelectric point is. But I probably will not give you an amino acid and say what is its isoelectric point. The equation of PI equals 1/2 pKa plus pKb. That equation, I'm going to say I'm going to reserve the right to have that there, but I will not ask you for a specific amino acid. If I ask you that question, I will give you pKa's. I'll say you have a new amino acid that has the pKa, C term is this, N term is this, or something like that, if I do that. Do we have to provide our ID for the test? I think you do, but like I said, since my account's not quite set up, I will get back to you once I know everything, and I'm planning to hammer that out on Tuesday. Anything else? Are we allowed to have a calculator? Yes, you're allowed to have a calculator. Yes, because we're going to do some calculations. I guarantee there's going to be at least one question that you have to do a calculator. You don't have a class for review on Wednesday. You have a regular class for material that's going to be on test 2. We have to know the C and terminus. So for amino acids, every C term has a pK of 2. Every N term has a pK of 9. I'm not going to have you memorize all 40 specific numbers because that's a pain. We're just going to say 2 and 9. N terminus is 9, yes. NH3 plus, COO minus. That's actually a very good way to remember. N is 9. Anything else? Well, if you do have other questions, as I do a picture of my dog, feel free to email me. We can set up a discussion. If you want to do like a video chat, I mean like a Zoom one-on-one meeting, we can do that. If you have questions you want to email me, we can do that. Otherwise, I will see some of you on Tuesday. Hopefully see the rest of you on Wednesday. So let's do that. Stop share. All right, I'll post this online. I'll post homework online as well. So take care, everybody.